Welcome to my channel Adhyan with Flow. So let's start nitrogen assimilation, guys. Now we were going to discuss about nitrogen assimilation. How plant use actually, yeah, this reaction we discuss further, guys. You have to understand first this thing. After you can betterly understand this reaction. Before starting this mechanism, I have to clear that one enzyme is most important. They play most widest role in nitrogen fixation, nitrogen assimilation. The name goes to nitrogenase. Yeah, okay. We know that nitrogenase, this enzyme is okay, important. Okay, we admit. What kind of things, what kind of compounds they contain? What is the structure of nitrogenase? They can easily make a ammonia from nitrogen. No, na, assimilation means nitrogen to ammonia form. We want nitrogen convert into our ammonia form. Ammonia form, nitrogen convert into our ammonia. Now, how they change? Yes, they start here and they complete here. Now they contain some metallic compounds like molybdenum, vanadium, and iron. When we discuss about micronutrients, macronutrients, we already discussed that nitrogenase enzyme contain molybdenum, nitrogenase enzyme contain iron. So here molybdenum and iron contain. Okay. Now any structure? Yes, guys. Yes, they have a structure. Iron made up of two subunits like this. Iron made up with two subunits. This is not in your NCERT, guys. I make you advance. Now another molybdenum. They made up with also subunits. And inside they also contain some iron groups. So iron is most important, guys. Molybdenum is most important. Here nitrogen bind. Here nitrogen bind. And here at the end product we got ammonia. And what is mechanism occurring here? What happened with this mechanism? What is happening here? So nitrogen change into ammonia. So this process we discuss here. Now this is the nitrogenase enzyme. Clear, guys? They contain molybdenum and they contain iron. This is the arrangement: molybdenum, iron, and iron arrangement. Clear, guys? Now they take what? They take nitrogen. So here nitrogen will bind. Nitrogen triple bond. Look at here. Nitrogen is bound. Yeah. Here nitrogen is bound. Yes, guys. Yes. Fine. So binding of substrate. Okay. So substrate bind with their enzyme. Now what's happened to next? Now reduction is here. Now the enzyme, what they do? They break one bond. Look at here. I'm zooming, guys. I'm zooming. Look at here. Here, three bond. 1, 2, 3. Three bonds now. Now what happened with next? During reduction process, one bond will break. Now look at here. Only two bond. One bond break, and these hydrogen ions means they are in this form. If one bond is break, then now structure is like this. Simple. Now this is not easy, guys. This is not easy. They take two hydrogen ions. They also take two electrons. They also take 4 ATP, guys. This is not a free of cost. Nana. Not a free of cost. Breakage of binding, they require this much. And again, that plant said, we need ammonia, guys. Move fast. Then what they do? They again break their bond. They break their bond. Now what happened? This is not still ammonia. Guys, this is not ammonia. Give me ammonia. But during this mechanism, another bond is break. Here is two bond and now here is one bond. So one bond also break. So they need two hydrogen ion. They need two electrons and they need 4 ATP, guys. So if we provide this much amount, then bond will be break. Now this time for ammonia. Guys. Now what happened? Next? Again reduction. Again breakage of bonding. Again we provide two hydrogen ion plus two electron plus 4 ATP. When we provide this much amount, then again this form is occur. Now we got ammonia. This is one group. This is another group. Means we get two molecule of ammonia. Here's look at here. Now after completion their function, enzyme will remove and product will remove. So this is the whole mechanism, guys. Now is clear. This bond also break, this joining also break, and they again need what? They again need this and this, guys. So now you can betterly understand this reaction. Now if we total calculate the amount, nitrogen, yes, nitrogen. 8 electron, we give 8 electron. Let's calculate, guys. 2, 2, 2 and 2. 2 electron, 2 electron, 2 electron, 2 electron. Total how much electron? 8 electron. Now 8 hydrogen, of course, guys. 2 hydrogen, 2 hydrogen, 2 hydrogen, 2 hydrogen, 8 hydrogen. 16 ATP we provide, yes, 4, 4, 4 and 4. Yeah, 16. So now you can understand. Now this form, now nitrogen with the help of electron, ions and ATP, they change into 2 molecule of ammonia. So now the process is done. Remaining part is H2 and remaining 16 ATP and some phosphate. So now you can betterly understand this reaction. Got it? Now got it. Little bit some description about nitrogenase. This is the extra. Yeah, guys, they made up with this units. Some extra. The enzyme nitrogenase is highly sensitive to the molecular oxygen. If oxygen is bind with the nitrogenase, then they get saturated and they can't do their function properly. It requires anaerobic condition. They work betterly in anaerobic condition. The nodules have adaptation that ensure that the enzyme is protected from the they made up. Nodules have adapt these kind of condition that oxygen can't enter in their nitrogenase enzyme. The protect these enzymes, the nodule contain an oxygen scavenger called lag hemoglobin. What they contain? They contain lag hemoglobin. They contain lag hemoglobin. If oxygen is still enter, they can take this. It is interesting to note that these microbes live as aerobes under free living conditions, but during nitrogen fixing events they become anaerobic. You must have noticed in above reaction that ammonia synthesized by nitrogenase enzyme. So this thing is complete. Now let's move further, guys. Now let's move. This is the rate of reaction, guys. What kind of reaction will happen? Ammonia change into amino acids. Ammonia change into amino acid. I'm introducing again from starting. I'll repeat. I'll totally recap all the thing one by one. Listen to me carefully. Guys, this is the root. Assume this is the root. This is the root here. This is the epidermis and cortex, endodermis, phloem, xylem, whatever. Now, root absorb what form of the nitrogen? Root absorb what form of the nitrogen? This form. And during nitrification, this form changed into then, and this form is toxic for plant. This form is toxic for plant, so they directly contribute to as a amino acid formation. This form to this form conversion, this form to this form conversion with the help of one enzyme. We call it as a nitrate reductase because we call it as a nitrate. And now this conversion to this conversion due to presence of one enzyme, nitrite reductase. And this conversion to this conversion, they happen normally, guys. They happen normally. And this mechanism, where this mechanism is produced, this is present in plastid. Where this mechanism is occur? In cytosol. The whole mechanism. Now, this whole mechanism is conduct. Yeah, I know that nitrate form absorbed by plants' roots, but they supply further now. They supply further to the shoot part of the plant. Then this reaction occur in leaves. In leaves, guys. Now this is the some fate of ammonia. How they change into amino acid? This is the amino acid, guys. Glutamate amino acid. What they happen? What happened? Reductive aminification. Alpha ketogluteric acid, ammonia, NADPS some energy change into glutamate. This is the amino acid. This is the enzyme. They convert into amino acid. This is the process, guys. Ammonia to amino acid. Ammonia to amino acid. How this ammonia? What is the fate of this ammonia? Now transamination. This is the amino donor and this is the amino acceptor. Look at here. Look at here. This form, yeah, this, but this is the donor, na, guys. So they donate their self, and this is the acceptor. Again, I'm repeating again. Wait. This is messy. Look at here. Here R1 group is here. Here R1 group is here. Clear? This thing is clear. Now the whole structure of this amino donor, it's switched towards this. Okay. Now another. Look at here. This is the R2 group and this is the R2 group. This group will switch accepted by R1. Look at here. So this is the transamination. There is one amino donor and one amino acceptor. How they produce? They produce two important amino acids, asparagine and glutamine, not glutamate. Okay, found in plants are a structural part of proteins. Yeah, this is the structural protein of the plant. Of course, guys, now ammonia change into amino acids, na, guys. They are the important structural and functional unit of protein. Ammonia bind. They make peptide bond and finally they produce polypeptides. Polypeptides, now their future is protein. Okay, guys. So that's all about your nitrogen assimilation. Nitrogen assimilation. So that's all about, guys. That's all about. So if you still not subscribe my channel, then please subscribe, G. Please subscribe. And thank you for listening me. And keep revise. There are not a two-step only. This is not a two-step only. There are huge steps, guys. Multiple step product. Your book given this, that's a two reaction. Reductive amination and transamination. There is huge. How glutamate further react with. And what they produce. Further, glutamate future is they produce what? Glutamine. This. Okay, and glutamine they further produce what? Glutamate. And glutamate, yes, they can also produce asparagine. So you know now there are huge complications, but your NCERT giving this two thing. That's it, guys. So that's all for now, guys. We will meet in our next video with a new chapter. Thanks a lot.